Now that we've learned about the taxonomic system, let's talk about the categories that we put things into. There are three. The domain is the largest category that we have now. There are three of them. Two of them are composed entirely of bacteria. Now, the old name for all bacteria was Kingdom Monera, and I put this on here strictly so that you'll recognize that name and know that it applies to prokaryotes. When I was in school, that we talked about Kingdom Monera, and you do sometimes still see that in the writing. Okay, so just so you'll know that when you see Monera or Monerans, it's referring to bacteria. But now we know a lot more about bacteria than we used to, and there are fundamental differences between the two groups of bacteria. One group is called domain Archaea. These contain very extreme organisms that live in very extreme environments. And the other is domain bacteria, which includes our common everyday bacteria that we come in contact with on a daily basis. So the two domains of prokaryotes are Archaea and bacteria. And the third domain is domain Eukarya, which includes the Protista, fungi, plants, and animals. So let's talk about the bacteria. Okay, there are two domains of bacteria because they differ in very fundamental ways. Okay, this wasn't known for a long time, but there are definitely some different kinds of bacteria. The common everyday bacteria that we know about are called bacteria. That's domain bacteria. The kingdom is Eubacteria, and they have certain sequences, certain characteristics. So ribosomal RNA, they have some unique sequences that are just unique to bacteria themselves, to the domain bacteria. They have one kind of RNA polymerase that is used to make messenger RNA. Their DNA does not have introns. Introns are extra pieces of DNA that have to be removed from the messenger RNA before it's translated into a protein. Those are not present in bacteria. The growth of bacteria is inhibited by antibiotics. When you expose them to antibiotics, it slows them down or stops their growth. Which is good for us when we get an infection. Their cell walls have a substance called peptidoglycan, which is a complex of protein and carbohydrate unique to Kingdom Eubacteria. And their DNA does not have any histones, those little proteins that the DNA kind of spools around or wraps around as it makes the chromosomes. Now, domain Archaea is a little bit different. It's also prokaryotes, okay? But there are RNA sequences. They have some that are unique to this group, but some of the ribosomal RNA sequences are like those that you find in eukaryotes. They have several kinds of RNA polymerase, some of which are very much like eukaryotes. They do have introns. Introns are, like I said, extra pieces of DNA that are in between the parts that code for proteins. They have to be removed when after the messenger RNA is transcribed before it can be translated into the protein. And this is present in Archaea and also present in eukaryotes. The growth of Archaea are not inhibited by antibiotics. They don't work on them at all because the antibiotics generally interfere with the formation of cell walls in bacteria, and the cell walls of Archaea are different. They do not have peptidoglycan like the cell walls of the bacteria. And they have histones with their DNA. Again, there are a lot of things about Kingdom Archaea that are more similar to eukaryotes than they are to the other group of bacteria. So let's talk about some characteristics of each domain. Domain Archaea are called extremophiles. They live in very, very extreme environments, very high heat, high salinity, high acidity, or very low pH or high pH, either one. Very, very different from what most things can live in. They're unicellular and prokaryotes. They do not have a nucleus. There's one kingdom in domain Archaea. It's called Kingdom Archaebacteria. Their DNA has both introns and exons. The exons are the parts that you put together to make the protein. They mostly live in extreme environments. They were probably the first organisms to evolve. They include things like methanogens and halophiles. Methanogens are bacteria that make methane gas. Halophiles are ones that like really salty environments. You find really salty environments like the Dead Sea, in really hot and acidic places like this is around Yellowstone, where you have lots of sulfur content and all kinds of things in the water that other things can't live in. So they're very, very unique in that they usually live in very extreme conditions. The second domain is domain bacteria. There's also one kingdom in this domain. It's called Eubacteria. These are also prokaryotes with no nucleus. as an identifying characteristic, they do not have introns in their DNA. All their DNA is transcribed and translated exactly as it is, so it's composed only of the exons. Their cell walls have the substance called peptidoglycan in there. An example of domain bacteria is E. coli. The full name is Escherichia coli, but we call it E. coli. This is a picture of E. coli. So it's a rod-shaped bacterium. And any of the bacteria that you usually come in contact with, like staph and strep and all kinds of other things like that, the ones that live on your body, in your intestines, in the dirt around your in your yard, and so forth, those belong to kingdom Eubacteria, domain bacteria. Domain Eukarya, all of these organisms have a nucleus. Most of them are multicellular, although some are unicellular. They have both introns and exons in their DNA, and there are 4 kingdoms that you find in domain Eukarya. We have kingdom Protista, which includes things like algae, seaweed, single-celled organisms that live in the water, and so forth. We have kingdom Plantae, which is the plants, kingdom Fungi, which is the fungi, and kingdom Animalia, which is the animals. Now let's talk about the kingdoms. All right, kingdom Archaebacteria, they are prokaryotic. They have cell walls without peptidoglycan. Some are autotrophs, some are heterotrophs, so they can be either one. Often they live in very extreme environment. They're called extremophiles. Okay, and examples are shown here. There's lots of different ones that are, there's a lot of them that have therm something in their name. That means they really like heat. Okay, methanobacterium, that means that they make methane gas. Okay, methanospirillum, again, that also means they make methane gas, and so forth. So very, very extreme environments, not the ones you normally come in contact with. Kingdom Eubacteria are the common bacteria. They are prokaryotic with no nucleus. They have cell walls that contain peptidoglycan. Some are autotrophs, some are heterotrophs, so they can be either one or the other. They're found in a variety of habitats worldwide, and examples, of course, are E. coli, as we mentioned before, Staphylococcus aureus, Staph aureus, that causes staph infections, and lots of other things that you come in contact with. Strep, all those different kinds are the normal kind of Eubacteria that you come in contact with on a daily basis. Kingdom Protista is what I sometimes call a catch-all kingdom. Okay, this is identified as being eukaryotes that cannot be classified as fungi, plants, or animals. Most of them are unicellular, but some are multicellular. Some are autotrophs, some are heterotrophs. There's a large variety of organisms in here. An example is amoeba, paramecium, all the different kinds of algae, seaweed, those kinds of things belong to kingdom Protista. They're very, very different than, they are eukaryotes. They do have a nucleus and other organelles, but they're not plants, animals, or fungi. There are some that are similar to plants, there's some that are similar to animals, similar to fungi, but they don't meet the characteristics for those kingdoms. So we put everything that doesn't belong in the other 3 into this one. In your book, you'll read that now the Protista is combined of several kingdoms based on various features, like whether they're animal-like or plant-like or so forth. We're not going to worry about that. We're just going to call them all Protista. Kingdom Fungi are the fungi. These are eukaryotes with incomplete cell walls made of chitin. Incomplete cell walls because the cell walls do not always completely enclose the cell. There are openings in them where they communicate with other cells. All members of kingdom fungi are heterotrophic, and they all absorb their nutrients that are digested outside their bodies by enzymes that are secreted by them. Most of them are multicellular, but there are some unicellular ones like yeast. And examples include mushrooms, molds, and yeasts. This shows a lot of different pictures of different kinds of fungi that you might have come in contact with, like puffballs and various kinds of toadstools and so forth. Kingdom Plantae are also eukaryotes. They are defined as multicellular autotrophs with chloroplasts and cell walls made of cellulose. All members of kingdom Plantae are non-motile. That means they can't get up and move around, although there is motion that occurs within the plant itself. All members of kingdom Plantae are photosynthetic. Examples, of course, trees, flowers, grass, all those kinds of things that you're familiar with from your lifetime experience. Kingdom Animalia are all eukaryotes also. They are defined as multicellular heterotrophs without cell walls or chloroplasts. Kingdom Animalia is the only kingdom that has no members with cell walls. Every other kingdom has members, at least some of its members, with cell walls. Most animals are motile in one way or another, whether they get up and move around or whether they have parts within them that move. Examples include all the things you see here, including insects, worms, mammals, fish, and so forth. and all of those are animals. Now, in modern day times, we use different methods to classify things. Nowadays, we use something called cladistic analysis. This is involved in making a diagram called a cladogram. This shows evolutionary relationships between organisms, so it kind of combines taxonomy and evolution together. It depends on the development of derived characteristics or the use of derived characters. These are new innovations or new characteristics that result in some kind of new branch in the direction of the cladogram. The derived characters are things that occur in populations, not individuals. It's related to evolution. Here we see a basic cladogram that has a frog, a lizard, and a rabbit. Okay, so if this is an ancestral species down here before you get to the branch, okay, now we have animals that have 4 limbs. All right, so we have here a frog. That's kind of a basic animal with 4 limbs. Frogs have to lay their eggs in water. And they undergo a metamorphic process from an egg to a tadpole to a frog. Somewhere in between the frog and the reptile, there was a development of something called an amniotic egg. This is an egg that is able to be laid outside the body away from water. So reptiles have the amniotic egg, so here's the derived character that separates them from the frogs. Now we have over here the rabbit. Well, sometime between laying the egg on the outside and having the eggs develop on the inside, that's where you have a development of that. So that's a new character separating the mammals from the reptiles. So that's just a simplified way to do it, and we'll do some activities in class to reinforce this. But that's the basics of cladistic analysis. Here's a cladogram. Okay, you can look at the taxa, which are the groups that we're looking at here, and the characters that they could possibly have. So you have the different kinds of characteristics and the different organisms that you're looking at, and so you group them by how many things they have in common. Okay, if we have a lancelet, which is a non-vertebrate chordate. We have a lamprey, which is a type of a primitive fish-like eel. We have a tuna, a salamander, turtle, and a leopard. Okay. All of them they're all chordates, but the lamprey, tuna, and salamander all have a backbone, so they're what we call vertebrates. So here's the derived character of the vertebral column. Right. The next thing we have is hinged jaws. Lampreys do not have a jaw; they have just an open mouth. But all the other animals have that open jaw, and so here the derived character is the hinged jaw. Next, we have walking legs. Now, fish have limbs, but they don't have walking legs. But all the other ones do, so here's the derived character of the 4 walking legs. Next, we have the development of the amniotic shelled egg, which separates the reptiles in mammals from that. And then the development of hair, which is the leopard, which gives us the leopard. So you can classify them based on the number of shared characters that they have and figure out how they are separated from each other and what kind of major characteristic changes happen between the groups of animals. This cladogram represents the 6 kingdoms and the 3 domains of life. We have somewhere in the past a common ancestor, domain bacteria, which has all the Eubacteria in it, Archaea here. Notice it's branched off closer to the eukaryotes because it has characteristics in common with eukaryotes. Then we have the Protista over here, single-celled organisms. We have the plants and the fungi, and over here the animals, and shows all of the different phyla of those groups within those kingdoms. So all of these diverge from a common ancestor in the far distant past at one point or other. More recent thinking has the Archaea and the Eukarya on a branch separate from the bacteria, and then there's another branch here. So that understanding and knowledge about this is still evolving over time as we learn more and more things about organisms.